We have a paired presentation out of Yale University School of Medicine. Yong-Hui Jiang and Dr. Jiangbing Zhou are going to be presenting to us on a novel gene editing approach for long-term paternal gene activation in Angelman syndrome. Welcome.
Great. So first, thanks, Alison, for your introduction. It's a great pleasure to be here. So in the past, I heard a lot about the foundation from Yong-Hui. Actually, this is my first visit to this FAST event. So I heard that this community have been working together work together to find a cure, to care each other. I also heard how well the FAST Foundation has been running. So by now, I was truly amazed and impressed. So we are a group of scientists at Yale. Probably is about more than scientists, because our Professor Yong-Hui Jiang is a leading physician in the field who sees and treats patients every day. So, but we share the same goal with all of you. So we want to dedicate our own efforts to find the treatment or cure for Angelman syndrome. So our approach is develop a therapy, use a genome editing approach. So thanks to the speaker in the morning, so we know that there are 3 different types of gene therapy, including gene replacement or gene augmentation therapy. So suppose Dr. Wilson is speaking here right now. He actually is talking about how gene replacement therapy is promising for Angelman syndrome. So you will hear that tomorrow morning. The second type is gene suppression, so which can be achieved through delivery of siRNA or like ASO. So the third type is genome editing. So in this case, one can deliver a DNA nucleus. So we're excited about all 3 different types of gene therapy approaches can be applied for Angelman syndrome treatment. So right now, we're particularly excited about the potential of ASO-based therapy, because we have ongoing clinical trial, which has shown positive signal. So we're excited because probably within a few years, we're going to have treatment for our children, our family, our friends. We're also excited as a scientist because we know that this treatment mechanism works, right? Because now we can suppress the antisense sequence, we can achieve treatment. But we also learn from these clinical trials, but actually not only from clinical trials, we can also learn from the biology. ASO therapy is promising, but has limitations. Mainly because ASO works on RNA level. So we learn from the morning, right, from DNA, RNA to protein. So ASO works on the RNA level, which means that regardless of how frequent your ASO or how many ASO you delivered, you always have this antisense exist, right? So with time, ASO degrade, and then the suppression just comes back. So how to overcome this problem? So the emerging technology based on CRISPR editing could be the solution. So how the CRISPR works, right? So this described in the morning presentation by a speaker. So CRISPR works by a machinery called RNP. Is mainly include a Cas9 nucleus and guide RNA. So the nucleus can move into the chromosome and cut DNA. So it's dangerous, the nucleus cut DNA without specificity, right? So that's why we need to have the guided sequence. So the beauty of the CRISPR-based genome editing is that, so once it's cut, because it happens at the DNA level, so the antisense sequence will be permanently eliminated, right? So one treatment should be enough. So that's a great promise of genome editing therapy for Angelman syndrome treatment. So CRISPR can be potentially delivered in 3 forms, either DNA form, or RNA form, or protein form. So as mentioned in the morning talk, right, CRISPR is dangerous because the nucleus is active, can cut, potentially cut the DNA without specificity. That's why we need to provide a guide. But in reality is that even without guide, this nucleus potentially can cut DNA. That's very dangerous because what if cut the normal gene, right? There could be problem. So to overcome this problem, the limitations or concerns, potentially we can deliver protein-based CRISPR. So the advantage is that the protein-based machinery, once get into cells, so the machinery will degrade within 24 hours. So basically hit and run. So instead, if we deliver CRISPR machinery use DNA or RNA form, the active machinery could be potentially exist in cells over a long time. That could be very risky. So therefore, we think probably the ultimate solution for genome editing-based therapy for Angelman syndrome might be protein-based CRISPR therapy. So the problem is that how to deliver the protein-based machinery into cells. So we know that viral-based vector cannot do that, because viral-based vectors always deliver DNA form. So to do that, potentially we can use particles, right? So when we talk about particles, everyone's thinking about lipid particles, because I assume all of you have received the COVID-19 vaccine. So they are delivered using lipid-based nanoparticles. So our group have been studying another type of particles, which are made of polymers. So single components. These polymer particles can be made through one single polymer. They can assemble into nanoparticles. Compared to lipid particles, they have certain potential advantages in terms of stability. For example, as shown in that figure, our particles are very stable, can be lyophilized, even can put on the room temperature for a long time. And also, the polymer particles can potentially deliver genes in high efficiency. So much greater, actually in the lab, and in the lab we know that they're much greater than most lipid-based systems, even the Moderna formulation. So for example, we also show that our particles can deliver protein-based machinery. Here we use ARNI-based report system to test our nanoparticles. So we deliver Cas9 protein together with two guides. So if delivery is successful, and the stop codon here, as shown on the screen on the left, will be removed. So then we'll see the RP, red fluorescent protein. So that means in normal conditions, these cells do not express red fluorescent protein at all. So if gene editing is successful, we'll see the expression of red fluorescent protein. So we found that our particles indeed can deliver CRISPR-based machinery very well. It can need efficient genome editing. However, we noticed that particles approach may not be viable for treatment of neurogenetic disease because we want to deliver genome editing into the brain, right? So our brain is quite unique because our brain is very dense in terms of the tissue structure. There are many different types of cells, many matrix protein. So as a result, the intracellular space in the brain actually is very small. So as shown here, limited to 60 nanometers, very small. But unfortunately, typical nanoparticles, including lipid particles or polymer particles, are quite large. It's about over 100 nanometers. So that's why this approach, although we have the technology, this approach may be a lot of work for Angelman syndrome AS treatment. So fortunately, in our toolbox, we have another tool we call STE technology. So it's a chemical engineering technology. So what we can do is that, for whatever payload, is a protein-based payload or nucleic acid, we can chemically modify our payload. For example, if it's RNP-based, Cas9-based RNP, we can put 2 or 3 molecules on the surface of our Cas9 protein, and then our machinery can get into cells through, non-endocytotic, which means that it can just penetrate cell membrane, get into cells. And after penetrating cells, all chemical modification just fell off. So the machinery will turn to the native form without losing any activity. So how this system works. So we show that this approach is very efficient in terms of delivery, as shown here. We know that this technology can be utilized for delivery Cas9-based machinery for genome disruption, which we will talk about in the next probably 10 minutes. We also show that same technology can be utilized for delivery of genome epigenetic editing machinery. We can also deliver protein, so whatever protein. Here we show the antibody. Of course, you can deliver enzyme or like transcription factors. So as long as we can do the chemical modification, we can deliver them. They can get into the cells and then high efficiently. We also show that this technology can be utilized for delivery of ASO or even DNA or RNA. So in terms of efficiency, I think it's much greater than most particle systems. And this, we have working in the particle field for over 15 years. So this technology is highly efficient, and most of the time more efficient than particle approach. But the most attractive of this approach is that after the chemical engineering, our payload actually are very small. It's typically 5 to 15 nanometer. So remember I mentioned in the brain, the space is very small. It's less than 60 nanometer. We cannot deliver large pharmaceutical agents, right, or particles. So those therapeutic agents, even after modification, only 5 to 15 nanometer is perfect for brain penetration or diffusion. And also another advantage is that unlike nanoparticles, remember, for example, when we receive COVID-19 vaccine, right, everyone receive probably 100 microgram nanoparticles. So within the 100 microgram, in reality is that you receive 99 microgram lipids. Those are carrier materials. So STG technology is different. We deliver 100 microgram therapeutic agents. We actually receive probably 98 microgram therapeutic payloads. The carrier materials is much less than the nanoparticle approach. So this approach has been very promising, particularly for treatment of neuro-based disease. So we were in this study, for particular Angelman syndrome study, we use first machinery as shown here. So Cas9-based machinery that are modified through the STE approach. So this is reagents which make our mice run so well. So a little bit more about the system. So as I mentioned, we use a protein-based machinery. So what it means is that we use Cas9 protein and then complex with the guide RNA. Again, the guide RNA provide guidance to guide our machinery into specific place. So we show that the resulting RNP is very small, about 12 nanometer. It's electric charge, or good for brain penetration. We tested the machinery in vitro and in vivo. In vitro in cell culture, we show that efficiently turn out the gene expression. And in vivo, we inject into the brain. We found that this machinery can edit the neuronal cells very well. And for whatever reasons, the specific machinery we choose, preferentially like neuronal cells. They edit the neuronal cells. So this is, of course, this is just local intraparenchymal injection. For Angelman syndrome, probably we don't want to do that. Actually, we did a RCV or intracerebral injection. So Professor Yong-Hui Jiang is going to talk about how to utilize the technology for Angelman syndrome treatment in animal models.
All right, thank you, Jiangbing. Also, thank you, Allison. And Allison was reminding me, said, you might talk a little bit history, and Jiangbing said that this is the first time. I said, oh, that's true. I'm sort of enough old for this audience, because when I was student, back to 25 years ago, our lab, our student, we discovered the UBA3 gene. At that time, I was the team. I look at it today, the 10 co-authors, I'm probably the only one still working on Angelman. Thank you, Allison, for keeping me up here. Also, many people recognize my name because when I was a student, I made the Angelman mice. And that time when I made mice, I did not know I was making Angelman mice, very frankly. When I started making mice, Angelman gene was not discovered. 1 year later, oh, I made the Angelman mice. So thank you for your citing my paper. That was one of my most cited publications in my career. So I'll give everyone credit for that. So almost like 5, 6 years ago, Allison invited me, and we were in the, I remember the first meeting was in Chicago, in the corner of only 4 tables. I'm one of 20 of 25 or 30 people at the first meeting. Clearly, like all of you, you impress Allison's leadership, skill. Probably I would say give her credit for the judgment. And then I also want to say all of you parents or grandparents all support for Angelman. That's make here like a 20 times bigger meeting than 5, 6 years ago. So I'm truly privileged to be part of this team and hope getting bigger and successful and can cure all the Angelman. So with that, I don't need to highlight too much more because a lot of talk already covered, just highlight a few. Compared to AAV, what's the new tool Jiangbing just described we are working on. And also when Allison and I saw, Allison saw our work, she said, Yong-Hui, can you do a talk here? At that time I was saying, okay, maybe Allison is too premature. But I always remember Allison asked, or any parents ask, bury or never say no to Allison or anyone family because that's the, because I see many Angelman or other patients, whenever everyone family come to asking if I can do something, I would say yes. Allison has a double head as a parent, also the CSO, I'll say yes to you, and let's work out the plan. That's why we are here today. So, so a lot of the comparison, which is Jiangbing highlighted already. So we truly think this tool probably have some advantage. I also recognize each tool has different advantage, and I'll say other tool has no disadvantage for. So, so this is sort of what we do. I will share with you some of this ongoing work, and then this is a design by one of research associate and Xiaobas Lewis. She's an audience here, which I'm lucky to recruiting her during middle of COVID. Extremely talented research associate. She's MD PhD. I also, when I'm recruiting her, I did not know she's an MD PhD receives extremely substantial kind of training for surgical training. We realized that's exceptional skill, make this project very successful. So I give all credit to Xiaobas for all the work she produced. I'm going to show next 5 or 6 slides. So the design was basically, you heard this kind of topic already. We have 2 lines. Scott Dindo, when he was in Arbodas lab, made that report line, many people using. Then first we're testing the system whether this new tool we have indeed working better in the brain cell. So we, as I said, Xiaobas was able to reliably inject intracerebral injection on day 1, day 2 pups. It's amazing look at how she does that work very reliably. Then we're first testing the system, the molecular efficacy. If you deliver P1, P2, then you check on about 30 days and 90 days, see whether reactivation happening. And the second, through the real Angelman mice I made 25 years ago to see indeed that reactivation can rescue the phenotype. So quickly go over some of the data. Clearly, the first thing you say, okay, does this deliver true with Cas9 protein IMP, chemical modified IMP? If non-chemical modified IMP, they can deliver all other organ systems cell very efficiently, but they were not able to go to the brain very efficiently. But with this one, you can see, based on the QRT-PCR, what you can show 3 different brain regions, prefrontal cortex, hippocampus, the cerebellum. You check, use the YFP, UB3 fusion protein. You also check the UB3 antisense. I will not go detail for this complex sort of diagram, but clearly you can see the UBC-YFP from paternal chromosome reactivated, then antisense in the opposite way reduced. That is corresponding very well. Before that, she testing like 4 guide RNA, multiple cohort and try, so this is sort of validation for that first. So we are pretty comfortable that reactivation is very robust. We also use the second method, which IHC, which you can clearly see. Oh, you can see this is the 30 days after single dose injection, 30 days later, and you sacrifice mice, do the staining, use the YFP, this look at the cortex, look at the cerebellum. This is definitely eye-opening when we saw this data. We will see that variability, but reach this level of efficacy, almost 70% of neuron injected, which is new end neuron, which is edited successfully. Similarly, a little weaker for the cerebellum, which you can see Purkinje cell. So that's pretty, to me, it's got very excited for this possibility. And the same thing, you can do the third method, complementary, which is doing the Western blot. This time, Xiaona did, which is, let's say, how a single dose, how long that effect will last. So you look at 30 days, looks good. Let's look at the 90 days, which you clearly see, you can see this is very, very clean, shows you after 90 days, that reactivation is still happening. Because if you don't have the treatment, it's almost no protein expression, after 90 days, you can see this is a wild type. So roughly you also say it's probably, we estimate about 40%, which is Allison told you 40% that should be make the kids, in the human, should make the human almost asymptomatic. So that's pretty good, everything. And then we're asking, it's a possibility whether this reactivation indeed can rescue the phenotype. Just keeping you in the perspective, rescue the mice is a much easier correct the human. Okay, keep in that perspective. So what I showed you in the mice, she has done multiple behavior assays, just convincing you the same way. Indeed, the reactivation, it's working. So first, it's looking at only 3 weeks after the injection, you already see the rescue for the open field testing. Normally, Angelman mice, maternal deficiency, they move less. After you treat, this is a pink one, you can actually make the mice moving more. And you use 2 different measurements. One is a travel distance, one is a center time indicated for the possible anxiety-related behavior. So both are working well, and then she actually repeat again, 8 weeks later, the same cohort, after 5-6 weeks. Because if you repeat too often, that got sort of habituation. Again, it's consistent and validated for this same behavior. And then the same thing, motor function has to be talking yesterday and today. This is actually, when I made the mice, this is probably one of the single most robust behavior phenotype in the mice. Almost everyone took my mice and they can reproduce this phenotype, even your first timer. So again, same thing, you can see this P1, P2 intrathecal injection I would say it's not 100% a rescue, it's a rescue pretty significantly for the single dose, both different paradigm, steady paradigm and accelerating paradigm, which you probably, there's a last video Allison showed you early in the morning. That was indeed this data from here, which is good to sort of both ways. You can visualize them or you can quantify them. Same thing, one of the weaknesses for the Angelman mice, not particular for the Angelman model, for many other neurodevelopmental mouse model, it's always a cognitive function. Even the human is very, very, very severe, like Angelman. We end up in the mice, you don't see strong, severe cognitive behavior phenotype like the human. So that's probably evolution difference there. That's speak for the value like Scott was given for pig and the monkey. Other probably have much more robust cognitive function phenotype. But this one we were able to yesterday, you also see we used the paradigm called novel object recognition, which has a few labs also replicate this one too, which you can clearly see, interesting to see. One is called short-term memory, one is long-term memory. Clearly they rescue the short-term memory pretty well. The long-term I would call probably partial memory rescue. And this one, Alice says, I hate this one, but I'm going to show it because she said, my Quincy doesn't do marble bearing. So, but which is in the mice, oftentimes we do it because it's easy to perform, which clearly you can see on the, it's not fully rescue for this particular behavior, it's a partial on that, but I think it is indicated just all complementary each other. We do see, I would say, it's not perfect rescue, but it's a pretty robust rescue for a single dose of an injection. And then, of course, the question comes to move in the human. Jiangbing already mentioned to you, we think IMP Cas9 protein is much better than the DNA delivery, also probably better than RNA delivery. RNA probably is also short, but the protein, we expect this protein machinery degraded in the 24 hours after going to the cell can reduce significant potential for off-target event. Of course, we don't know for sure until we evaluate at the end. But clear, but looking at the quicker for the acute toxicity, we don't see that, which quickly, if you use A9 line, the guard RNA, you look at the generalized sort of from the toxicity, liver function, the kidney function, it looks pretty within the, like a few days' time, look pretty normal. Then you look at the way that for this injected mice versus not injected mice are wild type, that's not much difference for the growth. And you look at the survival, also did not cause acute significant toxicity for the death. So that's all good indicator. I think my time is up, right? Then the last, of course, this is mice. As you know, mice and human, for the imprinting impression, it's very, very highly conserved. But at sequence level, they are not conserved. Therefore, anything you work on the mice will not directly applicable to the human. So you have to do it again from human. That's where we took the advantage for Dr. Wilson or Dr. Salko's paper early, and they identified some of the guard RNA. We say, okay, let's test this guard RNA in the iPSC cell line we generated. Thank you, fast support for this repository. So we're able to, this is early, but we're able to show at least one of the guard RNA in the human working pretty efficiently that we definitely need to validate this one future for the later time. This is only very early neuroprogenitor cell from iPSC cell differential neuroprogenitor. So that's what we are working on for the human. Eventually, we do need a human version of guard RNA for all the experiment or translational research for next step. With that, I'm going to thank you for all people working for this project for both lab. I mentioned primarily Xiaona is the person responsible for a lot of data she produced and working with the Ying from Jiangbing's lab. He is a chemist, kind of produced the IMP and the complex for us to test it. This is Jiangbing's lab during the COVID and on the Zoom meeting. Of course, thanks FAST for all the support of many, many years. And my collaborator for the Stem Cell Center or the other NIH and other. Jiangbing is the PI for NIH, so the somatic gene editing consortium. I'm the co-I for this consortium, so we took advantage for that support, make this technology happen. With that, we'll stop here. Thank you for your attention.